Okay, I'm wired up. I think everybody hears me. I'm Hugh DeLong again, in case you forgot from earlier today. And besides being the department head, I'm also the program manager for Natural Materials and Systems. I like to think of this program as the figure of merit free zone. Because I don't really think of these in terms of figures of merit. I think of it more in terms of trying to understand the process. So a lot of things we do is in the discovery area. In other words, it exists in nature and we want to understand how that works, okay? And so I stress that to all my PIs that whenever they are working on a particular system, that they must get to the point where they understand how it is happening in nature, because we'll never have the ability to mimic it if we don't understand what's going on. And so I actually don't continue projects if they don't get to that point, because that's, I think, is the most important thing. And so obviously nature has been doing a number of tasks over a very long time, and they've had lots of time to improve upon the process. So we're trying to understand some of those processes that we see in nature, and hopefully by learning how they accomplish the task, we'll be able to incorporate that into some future system in the Air Force. So I think about it more in terms of say, we wanted to study how the animals camouflage themselves, then we're interested in utilizing some of those techniques for camouflaging our own systems. But I don't think of it in terms of I have to be able to reflect a certain amount of light over a certain amount of time. So that's why I say I don't have figures of merit. So one of the things that I actually stuck on this slide just to point out this fact is this is a dragonfly. Because of the light shining towards me, you don't see it very well, but it's a very beautiful insect. This is a very brilliant blue. In fact, the second name to this species is resplendent. And if you hit this thing with light, it's actually, when the light comes down on an incident angle 90 degrees from the plane, you actually get reflection all through that spectrum. So any angle you're looking at this dragonfly, you're actually seeing that light. And so we were trying to understand, you know, how it accomplishes that, and it's all a matter of the built-in structure of the wings. So that's the type of thing that we're actually looking for. And then, of course, if you do understand these innovations that you see in biotechnology, these could be chemical sensors. I mean, biology has a very good ability of sensing things at very low levels, and one of the most important things that it's able to do is it's doing this in a very cluttered environment. So there's all kinds of other stuff out there. I mean, if you talk to DARPA and you talk to DTRA and you ask them about sensors, and they're always saying, you know, always have to worry about these other chemicals that are actually going to interfere and give you a positive. So they talk about false positives. Well, biology has to do with that every day because there's stuff out there all the time. So, but they're still able to accomplish very sensitive sensing that way. So we want to learn how they do that. Human monitoring is another option. And in fact, tomorrow when you hear Pat Bradshaw's talk, he'll get more into the human monitoring. I'm limited more towards the sensor area, and he's limited more towards what they're trying to learn for a particular aspect of the human and its operation. Covert tags and multifunctional materials for structural systems. You know, I just realized I don't have a laser pointer.
It's on the thing.
Oh, there it is. Okay. And so when you're thinking about implementing this vision, I've separated into looking at three different things. I want to protect the human assets. I guess I should use this thing. Protecting human assets in some way. So finding, defending against military significant threats. Looking at enhancing material performance, whether it's an existing material or whether I'm actually using the biomaterial because it happens to be superior. or enhancing system operations. Okay, so that's an example of that, mimicking nature's ability to find, track, and survive the enemy. And so these are the different science, or what I call research concentrates that I'm actually funding. And these are the programs that go along with that. And the way I've color-coded this is the green ones go with this circle, the sort of pinkish orange ones go with this one, blue go with this. And then, of course, when there's a blend, that means there's crossover, and these particular systems will actually enhance both of these operations. And so in the interest of time, I won't go through all those. But my program is essentially split into 3 sub-areas currently. There's actually going to be a couple of more added whenever they get into the system. But right now we're funding biomimetics. And in that case, I'm actually looking at the systems that I find in nature and trying to understand how they work and then trying to mimic those. The biomaterials are working with the actual material that you find in nature. Sometimes once we've learned how to utilize that, we may want to go into actually mimicking it, or we could use the biomaterial. I mean, there's a number of systems right now. You're wearing a lot of biomaterials because a lot of people wear natural materials like cottons. And then biointerfacial sciences. That's where I'm interested in the interfacial region, so it's biology to biology or biology to abiology, sort of like an interface. You find that in sensors all the time. It's also where I fund all my bio-nano or bio-meso systems, so that's where all the self-assembly is, whether we're building architectures or building arrays, the nano-combinatorics is all working in there. Okay, so this is the first area, and the first area is biomimetics. And so what I've done is I didn't put scientific challenges up here from all the programs that are involved in biomimetics, because I was trying to limit the number of slides I have. So I'm actually, I've just put a few down here to sort of give you a flavor of the type of programs that I'm funding under this area. And so biocamouflage fits in this area, and so we're trying to understand how the animal uses the various systems for camouflage. And so when you're looking at, say, an octopus or a squid, and you're trying to understand how the leucophores, the iridophores, the chromatophores are all working in conjunction with each other, and how those systems actually operate. And then we also want to understand how the animal actually controls them, because the animal has, you know, a ton of these all over its body, and it has to know how to turn them on, turn them off, how to make patterns. And so all of that is not understood, and you must understand that there's not a lot of neurological control over that. So that's another reason we're trying to understand is how does it go about accomplishing a task with very few nerve cells. So anyway, this would definitely make current camouflage ideas obsolete. The next one is chromophores. That tends to be a discovery program. We're trying to discover new chromophores in nature, so we have some people working on going out and actually finding new chromophores. The task also is to understand the mechanism behind that chromophore. How does the animal actually produce it? I mean, one of the major finds in that program is that one of my PIs discovered a new GFP, and it's the brightest GFP that's ever been seen. And so there's a number of people using that now because it's so bright, including AFRL, because they use it in the lab now when they grow up their cells as a marker, because you can literally grow these in cells, and it's so bright the light from it will reflect off the top of the Petri dish, so you can see it. Before, they used to have to open the cells to try to find the material in there because it wasn't very bright. Anyway, we're looking at new materials. We're also trying to get to some desired wavelength regions that you don't normally find in nature, so there's a bit of genetic engineering involved in some of that as well. Cell-directed assembly. There we're looking at getting the cell involved in building architectures, because the cell's outer membrane actually can organize materials, and so you can grow things out of the cell. And of course, we're also wanting to build things inside the cell. In fact, one of the things that, that deals with this type of activity will be next. And then finally, there are 2 lab programs that are involved in this area. One is Nancy Kelly, who's at RH, and she's looking at the RNA switches, and the other one, which are very fast switches, so that's why we're interested in those switches found in nature. And then Rajesh Naik, who happens to have a large program that's got multiple components, so you'll see his name multiple times in this talk. He's working on the chromophores, and also he's working in the iridophore area of biocamouflage. Okay, so now I have a science project, and what I've done is when I go into each area, I've got one science project that I've shown several slides of, so that you sort of get a taste of the type of science that we're involved in here. This one is called Bioenabled Particle Adherence for Integrative Spectroscopy. Essentially, it's looking at pollen and looking at diatoms, and we're interested in how they adhere to materials, because it's very important for pollen to be able to be transported from its starting point to its end point. And one of the things that pollen also has to do is, once it's been attached, it has to be able to release. And so we're trying to understand that mechanism behind that. And so the first thing we're looking at is understanding the biological approaches to that adhesion. And like I say, we're looking at different diatoms, different shapes, because different shaped diatoms will give you different adhesion properties. Sometimes they'll have these little spikes coming off of them, and so they add to the adhesion, and then here's pollen stuck to a bee. We also want to understand chemical approaches to mimic those. So it turns out that when you look at a pollen particle, it's not just those protuberances that you see sticking out everywhere. It's actually, there's a coating on there as well called pollen kit. And that actually is part of the mechanism of how this thing adheres. And then finally, and then also we want to understand about the structure of the diatoms, because we want to be able to manipulate that. And then finally, developing the chemistry of integration, because we also want to be able to adapt these for biosignatures, since that's an important aspect of the program. Okay, and so what I've done is, this is a broader team looking at different aspects. So I've picked out of it this one diatom project. And the reason that I picked this out is because it was actually a success that was previously a failure. Quite some time ago, we actually looked at diatoms, and we were trying to manipulate them to see if we could get them to build different structures, and it wasn't very successful. So it ended up not being continued. And now that we've gone into this program of looking at adhesion, the tools that are necessary to do that type of thing now are present. So it makes for a possible chance of being able to be successful again. And so the first step here was, I don't know if most of you recognize what this is. If you've ever been in a dentist office, you probably do. That's one of those sinks, you know, that's got the little hose in it and it's shooting the water around there. That's what it is. And so what they're doing is actually releasing the diatom, which is an algae, into the sink, and they're looking for the ones that stick to the surface. And then you just keep selectively selecting the ones that adhere to the surface, and eventually you get the ones that stick the best. And so this is the way nature does it, right? It's natural selection. You just keep selecting for the thing that gives you the best result. And so that's one way you can do this. Another way you can do this is by mutagenesis. So what you could do is just bombard the diatoms with ultraviolet radiation, what have you, and actually cause mutations to occur in the DNA. And then when these build the structures through biotemplating, you end up getting different effects. And so in the case of this one, you can see that whenever you build one of these frustrals, you have the two caps that are made, and you have one centerpiece, which is the girdle band that's joining the two. Well, in this case, when they did this, they actually got some that had multiple bands in the middle here. Also, you find some where you don't find the cap of the cap. In other words, it just made a ring. So that's another way to generate things that may be useful as far as structures. We're always looking for stable, inheritable mutations. Obviously, if it's a endpoint mutation, then the cell will die and you'll never see it again. So you're looking for something stable. And then thirdly, this is the project that we attempted before and was a failure. And now that they understand the genetic code better for this particular diatom, they were able to alter the genes and actually eliminate the genes responsible for certain structural elements, and were able to build these frustrals differently. So this is a wild type frustral. This one, by changing the gene, you were able to cause it to actually put an abnormal amount of silicon dioxide on there. And so the other option you could do is cause it to make these very silicon poor structures, and you can see this thing sort of looks like it's just draping over there because it's very thin. And so this leads us to believe that we could use the genetic makeup of this thing to actually make the structure that we want. Okay, the next area is biomaterials. And these are a few of the areas, and you heard that I actually fund silk. One of the reasons that we fund silk is we're trying to understand how to stabilize things in silk. It turns out you can put enzymes in there, you can put vaccines in there, and they end up being stabilized by being inside the silk. And the question is, how does that happen? So one of the things we're trying to do is study how that stabilizes the enzyme so that you no longer need refrigeration for this. I mean, you could imagine if you had an enzyme or a vaccine in there, you could just store it on the shelf in a climate like Africa where you don't have much refrigeration. Then you when you need to administer it, you just give them the patch made of silk, and they could eat it because silk is consumable. Silk, you can also understand how the structure of the assemble is changed by processing. So we're very much interested in biomanufacturing, looking at processing, and it turns out you can really change the properties of the silk fibers by the way you process them. And then finally, I have two different directions here. One is looking at peptide bonding to make materials, and that's Rajesh Naik. And then I have Wendy Goodson trying to tear them back down. So she's looking at how do you degrade materials with biofilms. And so the one I've picked, you guys, actually I get to be the first one to show this because I think maybe Pat might show this too. It turns out that Pat and I ended up studying the same shrimp, but for different reasons, because Pat's program studies vision, and it turns out that this shrimp has polarization vision. It actually sees circularly polarized light. And this shrimp also has a club. You can see it right here, that little lump. Turns out over time, the shrimp used to have just a spike, and it used to stab the prey in order to get at the fish that it needed to eat. But it also wanted to get at the crustaceans that are down there, so other crabs or oysters or what have you. And so it has evolved over time, that club. And essentially what it does is it beats the prey. And so it beats the shell. And the fascinating thing about this particular shrimp is that it throws that thing out at such a speed. It's measured about 45 miles an hour. And realize that's underwater. And so when it does that underwater, imagine you trying to take a rock to beat a shell underwater. You wouldn't be able to do it. But this shrimp can throw that out there, and it actually causes cavitation in the water, and so bubbles form. And so even if it misses the prey, the bubbles can stun the prey. So anyway, we're interested in why that works. Because when you think about it, this is chitin, you know, like any insect on land, and these crustaceans in the water, they're mainly made up of chitin and some mineral. It's mineralized chitin. We want to know why doesn't it break its arm? Why doesn't it break its elbow? And so that's why this is being studied. And it turns out that when the PI looked at this, it turns out it has 3 regions here. One's the impact region, which is very highly mineralized. It's very crystalline. They're oriented in 90 degrees to the plane of the surface. And so this takes the beating, essentially. And then this area here, which is the periodic region, has these sort of bands. And inside of those is a very mineralized fiber structure. And so that mineralized fiber can actually form cracks, and you'll see in the next slide, or 2 slides, how that actually helps. And then finally, the serrated region, which is on the outer edge, forms sort of like a girdle band on the outer edge. And so that's what this is, and you can see it really good here. It forms these layers that are very thin. I mean, it looks thick here, but they're actually thin, and they're oriented parallel to the surface. And what it does is it's like those. If you've ever heard of a ceramic bearing, and they put these iron bands around it to keep it from expanding so that the bearing holds up. Well, that's essentially what the shrimp is doing. This is around the outside of that club, and it keeps the club from deforming when the club comes in for that big smack. Okay, so how does it toughen? Cracks are actually nested in the periodic region, which was those, you know, you saw those periodic bands. And so and because of the way it's structured, it's probably harder to see here because of the light. But this is the actual structure, and this is the structure being modeled. You can see that there's actually layers of these chitin fibers, and then they're all mineralized, and they twist. So there's this twist in the layers. So you could see that sort of helical twist to it. It turns out that turns the cracks, and so it causes a very tortuous path for the cracks as they're inside that periodic region. And they end up dissipating a lot of energy from that. And that's why these things are actually holding up pretty well. And so the other thing that happens is there's pore channels through here. And so just like Les talked about before in the self-healing project that we funded at Illinois, where they were building all of those channels in the system, it turns out these have built-in channels already. And these channels actually, we'll have to talk about this sometime, these channels actually have their own pores that allow things to be distributed throughout the layers. And this goes all the way from the outside to the in. So we think this is how the animal may be repairing itself as well. So that is actually still under study, so that's more hypothesis than anything. And then finally, the last area is the biointerfacial sciences. So structural coloration is under there. So we're looking at things like you saw earlier in the first slide with the dragonfly. That's a structural coloration. And we're looking at new ways to make highly ordered biophotonic nanostructures. Also, the bio-nano-combinatorics is in there, more like combinatorial arrays. One of the interesting things about that is we've incorporated these mathematicians that are involved in learning, and we're trying to understand ways of learning how to smartly select what you should have in there from the first place, because whenever you're building arrays up, you have lots of possibilities. So you usually make these gene chips that have like 20,000 different combinations. And so instead of doing that, what you want to do is learn how to make smart choices from the beginning so you don't have to do that. It'll actually reduce your experimental costs as well. And then programmable materials, which is what we're going to go into now. And in this case, I have somebody from RY, Brett Winter, and he's working on whole cell microwing resonators. And Rajesh Naik is looking at the structural coloration, and he's also looking at catalysts, which I did talk about last year with the little ferreting balls that exploded. Those were actually pretty neat. Anyway, So this program is a MURI with Chad Merkin at Northwestern, and he's looking at taking nanoparticles, which we funded in the past, and putting DNA on them and essentially making these structures here and forming these lattices, similar to what you think about when you think about the lattices that atoms form, except these aren't at the atomic level, these are at the nanoparticle level, so they're a lot bigger, but they still give you the same properties. He's also doing, he has modeling going on with this as well, and they're looking at structure property relationships for all aspects of that. So that's whether the nanoparticle, whether it's the spacer, whether it's the attachment point, and how it sticks well, all driven by thermodynamics. And so this is the programmable atom equivalents, as he calls them. I think there used to be spherical nucleic acids before. But anyway, what they are is nanoparticles that are decorated with these different combinations of DNA. And essentially, as you decorate these with the first one, now you're putting on the second one to give you the linker, and these little light blue areas are actually how they're being linked to each other. And so there's a thiol-modified DNA, a duplexer strand, so this is the spacer region, that's the linker region, and then finally your sticky end is where you stick to other particles. That's a very important aspect because you don't want it too long, you don't want it too short, because you want that to be able to be thermally driven. And so this is a very important hypothesis. The most stable structure will maximize the number of duplex connections. So that's the way they build this. They build this in terms of how do I maximize the number of connections when I'm making those lattices. And so when they connect these together, like this shows that these things are pretty far apart. Actually, the particles here are actually linked either by DNA to DNA directly. What the hell's that? DNA to DNA directly, or you can actually put a particle in here that you then remove the metal and it becomes silent to the X-ray so that it's not involved in the properties. And that's how you can get these extended structures. And so the topic that I decided to pick up today is how do you go about making these three-particle structures here? So you can make these two-particle where you just throw them in a pot and essentially let them, you know, eventually assemble. But the question is, how would you go about getting one of these where you actually get these third particles exactly at these lattice points? And of course, you cannot just throw these in a pot and have this happen because you'll get lots of different combinations. So this actually requires a stepwise process where you actually initially go with the free particles and form what he calls the parent structure. And so right here, they're free. And then as you lower the temperature, this is the temperature down here, as you lower the temperature so you're past this inflection point, you don't want to be too far past. You want to be close enough that, you know, it has a chance to be a little bit non-sticky so that you get them to organize themselves because they have to organize in solution. And then they organize into this structure. Eventually, once you've got the parent structure, then you can continue to lower the temperature further till you get past the second inflection. And again, you're not too far away from it. You add the daughter particles, and those daughter particles actually go into those wells that are thermodynamically favorable. And so there is a lot of research behind this. I mean, if you looked at the paper that he just put out on this in Science, this one, it's not that many pages for this particular project. And in fact, if you look at it, it looks pretty simple. You just throw them together and poof. But it turns out there was a lot of work in this. If you look at the support material, it's like 3 times the size of the actual paper, because trying to learn how to build those structures around each one of those particles and learning how to affect the thermodynamics of those particles, and then how do you construct those pockets, and there was a lot of work and effort. Now that they know it, they can put this into a program which they've actually built now, and they can get to the point where they say what they want you know, what they're constructing. It will tell them back what they need. And so that's been one of the big pluses in this. And so I thought I was pretty fascinated that he was actually able to get the 3 particles together. And so these are the ones that he's built so far, these 5. You don't have to build something that's actually in nature. And so these 2 here, the perovskites, do exist. But these 3 here are all new. They don't exist in nature. And so there's no natural equivalent. And then I put this in there because I wanted to make sure that you realize that these aren't just, you know, 4 atoms or, you know, 4 particles thrown together and then another 4 on this side, and then that's it. You actually can grow crystals. This is, I believe this one was a 5 micron crystal. And so these are all of the, this is a TEM of it. You can see the particles all around, and so those things are actually pretty ordered in there from using the DNA. So he can get rather large structures on this. And so, in summary, we're looking at a broad range of these biological initiatives. So I only showed you 3 main ones as far as the science, and I talked about a few of them in the challenges. But there's a number of others. This is a broad program because we don't have a lot of biology programs. So I tend to be a little broader to try to encompass more areas. And so we're placing increasing emphasis to accomplish the tasks that we need in the area of biooptics. We're also looking at the composites with silk. We're looking at sensor arrays with the bio-nanocombinatorics. This was a new direction for us. And directed self-assembly, I have some new efforts in that because we're trying to understand the nature of the assembly and then to actually self-assemble these structures. Because you don't want to have to build this stuff at the nanoscale, right? I mean, that'd be tedious. You want to actually have it just assemble on its own. You just throw it in a pot and make it. And then, of course, the programmability is very important because you really want to just plug it in and have it go. And so I showed you the first genetic manipulation of a biotemplated structure. We talked about the toughening mechanisms in that biocomposite with the shrimp. And finally, we used DNA to actually create materials by design. And so that's it. So do you have any questions? Yes.
So very nice thought. In that diatom thing that you showed in the business, was that only due to the vortex?
That's only due to the water trying to wash them off the surface and they're sticking. And so it turns out that those frustrals, instead of being perfect cylinders, they started to elongate because it turns out the ones that are sort of in those oval shapes actually stick better.
There was no alteration of the surface property?
No. No, it was just by having them flow and only collecting the ones that stick, and then you grow them up and flow them again. Every time you grow them up, they keep wanting to select. That's natural selection for just that shape. But that's a tedious way, right? Because it takes a long time and a lot of growth. What you really want to do is just go in and manipulate it to do exactly what you want. So, any other questions? No? I get the award for finishing early. 8 tomorrow, everybody.